Hello guys, I am first year MBBS student. Today we are starting with the first topic of the protein metabolism, that is urea cycle. The urea cycles, we know that urea is the end product of protein metabolism, and the other name of urea cycle are the ornithine cycle and the Krebs-Hanslett cycle. We know this cycle, ornithine cycle, because ornithine, which is an intermediate in the urea cycle, gets regenerated back. So we are known this as a ornithine cycle, and this cycle is elucidated by the Hans Krebs and the Kurtz-Hanslett. So we know this cycle as the Krebs-Hanslett cycle. Now we start with the site. The urea synthesis occurs in the liver, and we transport this urea into the kidney for its excretion. This urea cycle is partly cytosolic and partly mitochondrial. The first two steps occur in the, this is 5-step reaction. First two steps occur in the mitochondria, and the next three steps are completed in the cytosol. So five different enzymes are required for the different 5 steps. And one more enzyme, that is known as N-acetyl glutamate, is also required, which is the allosteric activator of the enzymes carbamoyl phosphate synthase 1, CPS1. Now the urea, which is molecular formula is NH2C double bond O NH2. It has 2 amino groups. First amino group is comes from the ammonia, and the second amino group is come from the aspartate. Now we discussed about the cycle. CO2 condenses with the ammonium ion. This CO2 comes from the Krebs cycle, and the ammonia cycle, they condense in the presence of CPS1, carbamoyl phosphate synthase 1, and converted into the carbamoyl phosphate. Here 2 ATP consumption takes place. 2 ATPs are converted into the 2 ADP and the inorganic phosphate, and here the N-acetyl glutamate is required, which is known as the allosteric activator of the CPS1, which is another enzyme. One more enzyme is CPS2, which is used for the pyrimidine synthesis, and that is the cytosolic enzyme, while the CPS1 is the mitochondrial enzyme. This is the first step in the urea cycle. Now the carbamoyl phosphate, which is synthesized, it condenses with the ornithine. It condenses with the ornithine in the presence of ornithine transcarbamylase. Ornithine transcarbamylase and converted into the citrulline. This is the second step, and these both 2 steps are the mitochondrial steps. Now the citrulline, which is formed in the mitochondrial, has to be transported into the cytosol and by the special transporter system. Now the citrulline it condenses with the aspartate, and in the presence of the enzyme arginosuccinate synthase, and converted into the arginosuccinate. Here 1 ATP is consumed, and it converted into the AMP and inorganic pyrophosphate. Here 2 phosphate bonds are broken, so we have considered the 2 ATP consumption here. This is third step, and now in the presence of arginosuccinate lyase, arginosuccinate is broken into the fumarate and the arginine. The arginine, which is known as the immediate precursor of the urea, and the fumarate, it can be used as the connecting link for the TCA cycle. This arginine, in the presence of the arginase enzyme, liberated into the urea, and again the ornithine is regenerated. Now this ornithine is regenerated in the cytosol. It has to be transported to the mitochondria for further continuation of the urea cycle. Then by the special ornithine translocase transporter की help से, with the help of the special ornithine transporter, we have to transport this into the cytosol. This is the 5 steps of the urea cycle. Initial 2 steps are occurred in the mitochondrial and final 3 steps occur in the cytosol. Now we can discuss about the energetics of the urea cycle. As we have discussed, there is a 4 ATP consumption occur in the urea cycle. Initial first 2 ATP utilized in the synthesis of the carbamoyl phosphate. 2 ATP are utilized in the carbamoyl phosphate synthesis, and 1 ATP is broken down into the AMP and inorganic phosphate for the conversion of citrulline to arginosuccinate. So here 2 phosphate bonds are broken, so we have to consider about the 2 ATP. So total 4 ATP. Total 4 ATP are utilized in the Krebs cycle. Thanks. Now regulation of the Krebs cycle and the disorder of the regulation of the urea cycle and the disorders of the urea cycle will be completed in the part 2. Please give your feedback, and if you want the video on the another topic, then please comment me. Thanks.